Welcome everyone. In the previous lecture, we discussed about nitrate assimilation. Nitrate is converted into ammonia, whereas ammonium assimilation is a vital process in living organisms that involves the incorporation of ammonium ions into organic molecules, particularly amino acids. Nitrate assimilation is intricately linked to ammonia assimilation in the nitrogen metabolism of living organisms. This connection is crucial for the utilization of different nitrogen source. Ultimately, contributing to the synthesis of essential biomolecules like proteins and nucleic acids. So let's begin to discuss the ammonium assimilation. First, we will discuss about ammonium ion in brief. When ammonium levels become excessive high in living tissues, it can have detrimental effects on the overall health and functioning of both plants and animals. One of the critical consequences is the disruption of transmembrane proton gradients. These proton gradients are essential for fundamental biological processes, including photosynthetic and respiratory electron transport. In the context of photosynthesis, the process by which plants convert light energy into chemical energy, the transmembrane proton gradients are integral for the generation of ATP, that is adenosine triphosphate, a molecule that stores and transports energy within cells. Disruption of these gradients due to elevated ammonium levels can impede the efficient production of ATP, thereby compromising the energy supply for various cellular activities. Similarly, in respiratory electron transport, a process vital for energy production in both plants and animals, ammonium interference with transmembrane proton gradients can impair the synthesis of ATP during cellular respiration. This disruption has cascading effects on the overall energy balance of the organism. Furthermore, ammonium toxicity affects the sequestration of metabolites in vacuoles. Vacuoles are membrane-bound organelles within cells that play a crucial role in storing and regulating various substances. The interference with metabolite sequestration can disrupt cellular homeostasis and compromise the cell's ability to maintain a stable internal environment. Additionally, the disruption caused by excessive ammonium can hinder the effective transport of nutrients across biological membrane. Nutrient transport is essential for various physiological processes, including growth, development, and overall metabolic functioning. The impairment of this transport mechanism can lead to nutrient deficiencies, further compromising the health and vitality of the organism. In summary, the toxicity of high ammonium levels stems from its interference with critical cellular processes such as the disruption of transmembrane proton gradient, hindrance of metabolic sequestration, and impairment of nutrient transport across biological membrane, ultimately impacting the overall well-being of plants and animals. So, due to positive charge, the ammonium ions adhere to soil particles. Plant roots can uptake the nitrogen in the form of ammonium into symplasm by utilizing ammonium transporters situated on the plasma membrane. Elevated concentrations of ammonium inhibit the repression, or you can say the expression of genes responsible for encoding nitrate transporters. There are two categories of ammonium transporter in plants. One is the low affinity transporter, which are not saturated, and high affinity saturable transporter, which are non-saturable, may involve aquaporins or non-selective ion channels. High affinity transporters are encoded by the AMT-RH gene family. The Km values for ammonium transporter vary between 10 and 70 micromoles. Plant AMTs primarily act as ammonium uniporters and possess multiple membrane spanning domains. From this slide, you can see that ammonium moves from the soil to the root cell symplasm following the electrochemical gradient through AMT1, as you can see in the figure. Once inside the cytosol, some ammonium ion may convert to NH3 due to the alkaline pH, and it could enter the vacuoles as the membranes are permeable to ammonia. The entry of ammonia into vacuoles is facilitated by aquaporins known as the tonoplast intrinsic proteins, that is TIPS, which is situated in the tonoplast membrane. Since ammonium NH3 can bind to an H ion and convert to NH4 ion, it becomes trapped in the vacuole in the ammonium form. Gaseous NH3 can also diffuse through cell membrane, leading to the dissipation of proton gradients crucial for substance transport across membranes. This process may affect proton gradients across thylakoids during photosynthetic electron transport or across the inner mitochondrial membrane during oxidative electron transport. Proton gradients across the membrane are essential for ATP synthesis and the transport of ions and other biomolecules. Due to the toxicity of excess ammonia or ammonium ions for cells, animals have evolved aversion mechanism to avoid ammonia. The process of ammonia assimilation in plants is intricate due to its diverse sources of origin. These sources can be categorized as primary or secondary. The primary source involves ammonia production from inorganic nitrogen, including ammonium ions absorbed by plants, and ammonia generated through nitrite reduction or nitrogen fixation by bacteria in symbiotic association with plants. On the other hand, secondary sources pertain to ammonia derived from organic compounds during metabolic process. This encompasses ammonia produced from the oxidation of glycine to serine in photorespiration. The breakdown of nitrogenous compounds such as asparagine, arginine, and ureates, and the degradation and deamination of proteins and amino acids. Urea, a nitrogen containing compound, can be absorbed by organism and its decomposition facilitated by urease leads to the release of ammonium. Nitrogen fixing organisms such as certain bacteria convert atmospheric nitrogen into ammonium as part of the nitrogen fixation process. Microorganisms break down organic matter in soil or other environments, releasing ammonium as a byproduct of the decomposition process. Ammonia is generated at various locations within plants. The enzyme responsible for catalyzing ammonia assimilation operate under distinct cellular conditions. Consequently, the assimilation of ammonia involves different isozymes of these enzymes. Moving forward to ammonia assimilation in plant, we can say that it is a fundamental process that entails the transformation of ammonia produced through activities such as nitrate assimilation and photorespiration into amino acids. This conversion is pivotal for averting ammonium toxicity within plant cells. To accomplish this, two distinct pathways are employed. In the primary pathway, ammonia undergoes a sequence of enzymatic reactions wherein it is assimilated into amino acids through integration into the carbon skeleton of organic acids. Simultaneously, an alternative pathway offers an alternative route for ammonia assimilation, potentially involving different enzymes or modifications in reaction steps compared to the primary pathway. The overarching objective of this pathway is the synthesis of amino acids, essential components for protein formation and integral to diverse cellular processes. Ammonia assimilation plays a critical role in nitrogen metabolism, ensuring efficient nitrogen utilization for the synthesis of vital biomolecules. The regulation of this process is intricately managed to adapt to environmental variations and maintain nitrogen homeostasis. The swift conversion of ammonium into amino acids acts as a protective mechanism, shielding plant cells from the detrimental effects of elevated ammonium concentrations. In essence, ammonia assimilation stands as a key biochemical strategy employed by plants to harness nitrogen efficiently and sustain cellular health. So, as I have already told that there is a two pathway, one is the primary pathway and another is the secondary pathway. So, now we will take up first the primary pathway of ammonia assimilation in plants. There are basically two enzymes that play the pivotal roles. One is called as glutamine synthetase and another is called as glutamate synthase and also known as GOGAT. GS is basically available in two forms within plant cell, one in the cytosol and the other in the root plastid or shoot chloroplast. Whereas glutamate synthase, that is also GOGAT, or glutamine 2-oxoglutarate aminotransferase is responsible for the transfer of an amino group from glutamine to 2-oxoglutarate. So, now we will discuss about the GS activity. Glutamine synthetase combine ammonium with glutamate to form glutamine. This reaction requires the hydrolysis of one ATP and involves the participation of a different cation such as magnesium ion, manganese ion or cobalt as a cofactor. Within plant, there exist two classes of glutamine synthetase, with one class located in the cytosol and the other in the root plastid or shoot chloroplast. The cytosolic GS forms are expressed during germination or in the vascular bundles of roots and shoots, contributing to the production of glutamine for the transport of nitrogen between cells. In contrast, the GS in root plastids generates amide nitrogen for local utilization, while the GS in shoot chloroplast plays a role in the reassimilation of NH4 ion generated during photorespiration. The expression of plastids forms of GS is influenced by light and carbohydrate levels, but these factors have minimal effects on the cytosolic form. This intricate regulation allows plants to efficiently manage nitrogen metabolism in different cellular compartments based on environmental and metabolic conditions. So, increased concentration of glutamine within plastids positively influence the function of glutamate synthetase, also referred to as glutamine 2-oxoglutarate aminotransferase, or in short it is called as GOGAT. This enzyme facilitates the transfer of amide group from glutamine to 2-oxoglutarate, resulting in the production of two molecules of glutamate, as you can see here in the figure. Plant possess two variants of GOGAT. One accepts electron from NADH, while the other accepts electron from ferredoxin. This figure shows the GS-GOGAT pathway, where both GS and GOGAT enzyme are responsible for the conversion. This biochemical process requires a reduced cofactor of the reaction with ferredoxin, serving as the essential cofactor in green leaves during photosynthesis, and NADH playing a similar role in non-photosynthetic tissues. In green leaves, where photosynthesis occurs, ferredoxin is utilized as the electron donor in the reaction. On the other hand, in non-photosynthetic tissues that do not engage in photosynthesis, such as roots or other non-green parts of the plant, NADH serves as the necessary electron donor for the GS-GOGAT pathway. This pathway plays a central role in nitrogen assimilation and amino acid synthesis, contributing to overall metabolic processes and growth of the plant. So, here we can differentiate the different types of GOGAT that we have already told that there is a two type of GOGAT. One of them accept electrons from NADH, and that is called as the NADH GOGAT, while the other type accept electron from reduced ferredoxin and called as ferredoxin GOGAT or FD GOGAT. So, the NADH variant of the enzyme is known as NADH GOGAT, is situated in plastids within non-photosynthetic tissues such as roots or the vascular bundles of developing leaves. In roots, NADH GOGAT plays a role in assimilating ammonium ion absorbed from the rhizosphere, the soil close to the root surface. In the vascular bundles of developing leaves, this enzyme is involved in assimilating glutamine that is translocated from roots or senescing leaves. On the other hand, the ferredoxin-dependent type of glutamate synthase, referred to as FD-GOGAT, is located in chloroplasts and contributes to nitrogen metabolism during photorespiration. The protein abundance and activity of ferredoxin-GOGAT increase with higher light levels. In roots, especially under nitrate nutrition, ferredoxin-GOGAT is present in plastids, where it likely functions to incorporate glutamine produced during nitrate assimilation. The reduction of ferredoxin in roots is facilitated by electrons generated through the oxidative pentose phosphate pathway in plants. As illustrated in this figure, the NADH-GOGAT is found in the non-photosynthetic tissues, including roots and vascular bunding of developing leaves. In roots, NADH-GOGAT plays a crucial role in assimilating glutamine generated from NH4 assimilation, whether absorbed from the rhizosphere or fixed by bacteria in symbiotic associations. Additionally, NADH-GOGAT in vascular bundles is responsible for assimilating glutamine, which is either translocated from roots or obtained from senescing leaves. The enzyme FD-GOGAT is situated in chloroplast and plays a crucial role in catalyzing the metabolic process of glutamine. This glutamine is produced as a result of the assimilation of NH4 ion generated during photorespiration, a key aspect of plant metabolism. Notably, FD-GOGAT is responsible for 95 to 97% of the total GOGAT activity in leaves, underscoring its predominant role in metabolizing glutamine in leaf tissues. This activity is crucial given that glutamine is produced from ammonia assimilation arising from both primary sources such as the reduction of NO3 ion and secondary sources as seen in the context of photorespiration. In terms of genetic regulation, most plants have a single gene encoding FD-GOGAT. However, Arabidopsis stands out with two genes that is called as GLU1 and GLU2 encoding ferredoxin-GOGAT, while NADH-GOGAT is encoded by a single gene, that is GLT. Both ferredoxin GOGAT and NADH GOGAT exist as monomeric proteins, emphasizing their distinct roles in the metabolic processes of glutamine within plant cells. The presence of these enzymes and their genetic regulation highlight the intricate mechanism that plants employ to efficiently manage nitrogen assimilation and metabolism. So now we end today's lecture based on the ammonium assimilation. In the next lecture, we will discuss the alternative pathways for ammonium assimilation. Thank you.